Okay, now we have discussed so far the DNA damages and the mutations arise from these DNA damages. Now we will look at the once there is the DNA damages, those DNA damages are repaired by a system in the cell. That system is called DNA damage repair system. So in this class we will discuss about the DNA damage repair systems also. So the first we as a recap we have to visualize again. In the last class I discussed it. So when there is a DNA damage, how we come to know that there is a DNA damage? We come to know that there is a DNA damage by the change in the behavior of the cell. The cell itself show us that there is a DNA damage. That's what we call the manifestation by cells, manifestation of the DNA damage by the cells. So the cells manifest the DNA damage. How they manifest the DNA damage? Through a change in their cellular behavior, and those behaviors are like these. Number one, the arrest in cell cycle, cell division, and this results into cellular senescence. That is the one response, one manifestation of the DNA damage. That there is a DNA damage, one manifestation is the cellular senescence. And because of the reduction in the cell division, there will be reduced growth, there will be reduced reproduction, and then when there will be reduced reproduction, there will be reduced rate of natality. Natality means the birth rate. And all these things will happen in response to the manageable DNA damages. When there is a DNA damage and that DNA damage is manageable, the cell can manage it, cell can repair it and then manage it and then remove all those damages, then be healthy and survive. Second thing is necrosis. That is another response by the cell. That's how the cell manifests that there is a DNA damage. Necrosis is the sudden death. Necrosis is the sudden death. Without bothering about anything, it will be sudden death. And that happens when the DNA damage is very high and it is not manageable. The cells cannot repair at all. Or even if the cell want to repair it, the cost to repair is too much in comparison to the benefits after the repair. Next is the apoptosis. When the repair is costly than benefits. So what happens when the repair requires certain cost, certain energy and times, and if the cell realize that there is no much benefit after the repair, the cell decide to kill itself in a programmed way. That is called apoptosis. So sometimes that's why we also call them programmed cell death. Anyway, that results all these things, necrosis and apoptosis, both of them lead to the increased cellular death, aging, mortality and reduced growth. So both of these two results in a reduction in growth, and both of them will also result in a reduction in the survival. The growth will be reduced as well as the survival will be reduced. Third thing is the appearance of the mutants and mutant phenotype. So if the DNA damage is manageable, the cells try to repair it, and after the repair, sometimes the repair is very perfect, very efficient, and all the deleterious damages are completely removed, and in some cases some errors are still left, and because of those leftover errors, the cells generate mutants, and those mutants will have a mutant phenotypes. So that is the one thing. And the fourth is the cancerous cellular behavior, or the formation of cancerous cells, and generation of the cancerous cell. This is a kind of like this one. It is like a mutant. The cancerous cells is a kind of mutant cells which arise due to faulty or maybe failure of the repair system. either there is a failure of the repair system. Because of that, the mutagenic effect, the effect by the mutagen or the DNA damage accumulates inside the cell, and the cell survive with those accumulation damages. And or if the cell try to repair it, but the repair is faulty, and because of the faulty repair, some damages are unrepaired, or even the repair system itself create new damages, and those new damages, like erroneous one, like a repair system which is erroneous one, and because of this erroneous repair system, it incorporates permanent modification in the DNA, and that permanent modification of the DNA will have the continuous or permanent propagation of the mutant gene, and that results into the generation of the mutants, and these mutations can result into the cancer also. So basically those mutations are called oncogenic mutations, and those agents which result into these kind of mutations, they are called oncogen. They are called what it's called? Carcinogen. They are called carcinogen because they result into the cancer. So basically all carcinogens, if we say all carcinogen are mutagen, but not all mutagen are carcinogen. But certainly the carcinogen has to be mutagen. Carcinogen has to be mutagens because they have to induce the DNA damage in the beginning. If they get repaired or they do not cause the cancerous type things, so what will happen in this kind of mutations, rather than causing like a cell cycle arrest, they promote the cell cycle. They promote the cell cycle and indiscriminate division of the cell, and that's result into the cancer. So if you look at this cartoon, that says the same thing, that there is a DNA damage either by the exogenous or endogenous factor, and if the cell try to repair the damaged DNA, if the repair is efficient or the damage is repaired or removed by the DNA repair system, the cells survive and remain healthy. However, if the DNA damage is unrepaired or it is repaired with some error in the cells, there will be continuous one by more and more damages will accumulate in the genome, and that will result. So here is like a DNA damage and the repair system is faulty, or is like a failure of repair system, or faulty repair system. Failure of repair system or faulty or erroneous repair system. So it will result into, as I already discussed, cellular senescence, apoptosis, necrosis, and that will result into the growth, survival, colony formation in the case of bacteria, growth kinetics. They all will decrease. And if there will be certain mutations, and that mutations will promote the cell survival, the cell proliferation, cell cycle, so that will result into the cancerous. So this will be like a repair system. So it will be like a error prone repair system. Because of error prone repair system, so this will result into the some mutations, and that mutations will result into the cancerous cell formation. Similarly, there can be another things. So basically the cancer cells are mutants. So there is also non-cancerous mutants. Non-cancerous mutants, that could be another effect, non-cancerous mutant, and these will be the cancerous mutants. They may also arise because of the faulty repair pathway. Now next thing is how we can visualize DNA damage and DNA damage repair experimentally. So this is the experimental visualization of the DNA damage and DNA damage repair. So basically what we do, we do experiments with the E. coli, and we use the E. coli as a model organism, and that is very easy organism to investigate and study these kind of questions related to the DNA damage and DNA damage repair system. So how we do it basically, this is very basic zero level key information that every student should have, so that you can have the better sense of the the particular better sense and maybe the logical sense of the understanding of this DNA damage and repair system and the pathway also. So basically how we do, we culture the bacteria, and we culture bacteria a normal wild type bacteria of the normal wild type phenotype, and we can also culture mutant strands also. So basically let's imagine first that we have a wild type E. coli culture, and let's suppose these are the LB tubes, or it can be flask also. So the culture could be in flask also, in the LB tube also. So what do we do here? So this is the one reference culture. This is your reference culture. And the reference culture, what you do after the culture, you don't expose to the UV. So basically we use UV radiation. Why we use UV radiation? Because this UV radiation was at the moment in the history a standard way of generating mutations and mutants. So it was a standard way of generating mutation in microorganisms, UV radiation. So in the reference culture, we do not expose the cells to any kind of UV radiation, and these are the test cultures. In the test cultures, we have increasing dose of UV exposure, and that you can do. You can increase the dose of the UV radiation by increasing the intensity of the UV radiation, or you can do the same thing by increasing the time of exposure. Like you keep the culture for 5 minutes and you keep the culture for 10 minutes. So the culture which has been exposed in the UV for 10 minutes, that will receive the more dose of the UV radiation. Okay, now what we do after this exposure or before the exposure in the reference, you plate on the LB agar, you plate the cells on the LB agar, and then you put in the incubator, overnight incubator. You come on the next day after 16 to 20 hours, then you look at the colonies in the LB plates. Look at the colonies here, for example, in this picture. So these are the, let's imagine these are the LB plates, and these dots are the colonies of the bacteria colonies. Okay, so now what you do, you count these colonies. So these colonies are basically manifestation, or they are the synonym with the growth. Okay, so you count these colonies and then you plot these colonies. So the number which you get from the reference culture, let's suppose this is whatever number. So here you will get the N0, or maybe you can say NR reference. Then here you get in the test culture T1, NT2, NT3, NT4. Means the number of the colonies in the test plate 1, test plate 2, test plate 3 and test plate 4. So what you do, you divide NR by NR. So whatever you get, definitely you will get 1. So that you call 100%, and then you divide NT1 upon NR. So that's whatever you get, that is called percent survival. For example, if you have like a 500 colonies and here you get 250 colonies, then 250 upon 500 colonies will give you 0.5. So that 0.5 will be called as a 50% or 0.5 survival. For example, here you see in a graph, this is 1% survival, 10% survival and 100% survival is a log table. On the log table it has been mentioned like this, the 1%, 10% and 100% survival. So what you do here, so this is your reference culture, so you put a reference culture that this is my 100% survival. Then here let's suppose you get 95% survival, here you get 80% survival, here you get 70% survival, here you get 50% survival, and so on. Okay, so what do you do at the different UV exposure? So you plot all these values against the UV exposure on the x-axis. You plot this percent survival here. So you may let's suppose dot come here, another dot come here, another dot come here, another dot come here. So you connect all those dots, you get this curve. So this curve is called percent survival curve under a mutagenic presence, under the or maybe in the presence of a mutagen. Here is the mutagen is the UV radiation. And it can be some chemical also. For example, ethyl methyl sulfonate. You can add ethyl methyl sulfonate in the medium, different concentration of the ethyl methyl sulfonate or a different time of exposure in that chemicals, and then you can do the same experiment and you count these the colonies, get the percent survival, and then plot those percent survival. You will get a curve like this. This is called the percent survival curve or the DNA damage effect. DNA damage effect, the effect of the DNA damage in the presence of some mutagen or physical or chemical. Next, what you do now, what is here is the same thing. So but it is not a wild type culture, but it is some like a mutagen sensitive culture, mutagen sensitive culture. So it is like a mutant strain that is DNA damage sensitive. How we know that is the DNA damage sensitive? Like we have any mutant strain, we don't know exactly, but we have a mutant strain that has some mutant phenotype, so it is different from the wild type. So we take that mutant strain and we do the culture, and when we do the culture and we repeat the same experiment, that this strain, from the strain, this is for the wild type, this is the observation from the wild type. Similar experiment what we do here with the mutant strain, and we do the same experiment and we collect the data from the mutant strain. When we plot, then what we find for the wild type cell, it gives a DNA damage response like this, and the mutant strain gives a DNA damage response like this, yeah, mutagenic effect response like this. So based on what we see here, there is a gradual reduction in the survival of the cell in the presence of the mutagen. But this is for the wild type. This is for the wild type, and this is the mutant strain. Mutant strain. What we see about the mutant strain, that the wild type gives the gradual decrease in the survival, while the mutant cells gives a very acute or maybe very steep or sudden decrease in the survival. Means it is highly sensitive to the DNA damage or the presence of the mutagen. So in this way, the scientists were able to categorize different strain of the virus as the like a damage sensitive, DNA damage sensitive and DNA damage resistant, or the mutagen sensitive and the mutagen resistant cells or strains. So in this way we can visualize, we can see what might be happening inside the cell. Or we can also visualize the extent of the DNA damage. We can say in this cell the extent of DNA damage is not that drastic or that deleterious or not that serious, but in this particular strain of the bacteria the DNA damage is highly serious or deleterious. So many things we can learn from these kind of experiments. Now after these exercises, when the scientists were doing so many things, and by accidentally the scientists discovered something, and that something was that the cells are able to repair a deleterious of radiations or the mutagens. So from this the scientists concluded that since the cells are able to repair or overcome the deleterious effect of the mutagens or the radiations, there must be some kind of repair pathway which will repair the DNA damages which are done by these mutagens or the radiations. So from there the scientists started to think that inside the cell there is some kind of repair machinery or repair system. So how this first discovery came? So this first discovery is called light dependent DNA repair or enzymatic photoreactivation repair, EPR, or simply light repair or photoreactivation. So these are the four names for the same repair pathway or same repair system. So this is the first discovered DNA repair system, and it was discovered by an accident. So there was a scientist, Kellner. He wanted to generate mutants of the fungus Streptomyces graeus through UV radiation. And as I discussed, the UV radiation was the common method to generate mutants. Why they wanted to generate this mutant fungus? So they wanted to produce this mutant fungus which can produce new and more effective antibiotics. So the Kellner exposed E. coli and the fungus on agar plate to various doses of UV radiation in order to calibrate his experimental system. During this process, he was frustrated that he obtained survival yields that were seriously irreproducible. Means every time he was getting different result. Every time it means, for example, this is a bacterial culture. Let's suppose this is a bacterial culture. Okay, this is a reference culture and this is the test culture. Okay, so this is the reference culture, this is the test culture. Let's suppose this is a wild type strain and this is the wild type strain culture. So what we do here, we give exposure, and here we give, let's suppose, UV exposure of a dose X, So let's suppose this is experiment 1. So what we do here, we get the colonies. Okay, so what we get the colonies, and then we count these colonies and then we plot it. UV exposure, and here will be your percent survival. So remember, the wild type survival will be called as 100%. And then next, what we will apply this one, let dose X, dose Y, and then dose Z. Okay. Sorry. Yeah, but dose, let's suppose dose X. Dose X is here. And so what happened in a dose X? Sometime we have, survival like this. Sometime we have a survival amount like this. Somewhere we have a survival percentage this. Sometime we have a survival percentage this. Many readings. So let's suppose this was the experiment 1. In the experiment 2, experiment 3, experiment 4, and experiment 5. Experiment 5. Or maybe this is experiment 4, this is experiment 5, and this is experiment 2, and this is experiment 3. So what we see here, in one experiment the survival curve goes like this, another experiment the survival curve goes like this, another goes like this, another goes like this, another goes like this. So basically in science, when we do some experiment and when we repeat the same experiment on another day, it should give the same kind of result. And if we get a different kind of result, a variable results, so then we call such kind of experiments are non-reproducible. So unless any experiments gives consistently reproducible results, we cannot conclude conclusively on a single statement. We cannot conclude anything conclusively. So the scientist was frustrated that why it is happening. So he realized after many things, he tried to troubleshoot, and after troubleshooting many things, and some removing out some variable factors, he realized that it is the visible light, fluorescent light in the laboratory as the main culprit that is creating these variable results. So he found that probably these light has some effect on the variable survival of the bacteria. When it has some effect on the variable survival of the bacteria, it means it must be interfering with the DNA damage response. So in some cases it is promoting, so it means there could be some mechanism which can interplay with the DNA damage system, or which can try to repair the DNA damage. So what he did, means, okay, now we will have one experiment. In one experiment we will try to not expose the cell with the visible light, and in another experiment we will try to expose the cell with the visible light. And that what will happen. First he discovered that the visible light is playing a role in this variable results. So it means the is a factor in this particular DNA damage biology. Similarly, here there was another scientist, that was Renato Del Becco, in the lab of the Salvador Luria, and then he also came to the same experiment when he was working with the bacteriophage lambda in bacteria. And what he found that when the UV radiated phage infected cells are exposed to the visible range of light, they produce more plaques. We will come about this one in the in the next class, basically. So anyway, from here these two scientists discovered that the light plays a role, and then what they did many experiments, and from their experiments they came to know that the light has a protective effect. That it has a protective effect and it repairs the DNA damages which are generated by the UV radiation or some chemicals. And because of the light exposure, the survival of the cell increases. So basically what they did many experiments, the similar kind of experiment like you see here. The same kind of experiment we have already discussed. This will be a wild type cell. Here will be no exposure. This will be your reference culture. Whatever colonies you will get, that you will call it like 100% growth, yeah, 100% survival. And these colonies which you get from the exposure, different variable exposure of the UV radiation, and then you plot it, then you will get the survival curve. So in the next experiment, what they did, so these cells, all these cells after this UV exposure were not exposed to visible light. They were not exposed to visible